Good morning, my dear students. I would like to teach you about this chromosomal structure. Regarding DNA and genes, we have discussed a lot in the previous class. Now, I will explain you the structure of chromosome. Every living organism is made up of cells. So, the human body, here an example I have taken a human body. Human body, the whole body is made up of cells. And each cell has got various organelles present in it. Nucleus, endoplasmic reticulum, ribosome, mitochondria, etc. All these are present in the cytoplasm. And the cell has got the very important controller of the cell is nucleus. And this nucleus have got chromatin threads inside. The chromatin reticulum we say. And this is in a thread format. It becomes condensed and thicker and become chromosome during the time of cell division. When the cell is not dividing, it is in a form of chromatin thread. When the cell is dividing, when the cell completely divides and forms two cells, isn't it? 1 into 2, 2 into 4, likewise the cell get multiplied. So during the time of cell division, this chromatin thread will become thicker and condense and forms chromosomal structure. About this chromosome only we are going to see. And this chromosome have got DNA and which contains the genetic material. So again I am telling you, these chromosomes are visible during the cell division and it contains the gene, the genetic material, information which is carried from one generation to the other generation. That is from parent to child. So again the same points are repeated here. The human body is made up of millions of cells. Each cell contains nucleus and the chromatin network or coiled fibers which are chromosomes, and this chromosome are visible during the cell division, and this chromosome are highly condensed thread-like structure. DNA full form deoxyribonucleic acid. This DNA is present in the chromosome and this DNA contains the genetic material. Genetic material is nothing but the hereditary information which are carried from one generation to the other generation. So, we are going to see about chromosomes which are in a highly condensed thread-like structure which is present inside the nucleus in a form of chromatin network, and this becomes thicker and condensed and forms chromosome. This chromosome is visible only during the time of cell division. And already I told you, genes or the segments of DNA which is found in the chromosome, and this DNA is responsible for inheritance of a particular phenotype character. So the DNA is responsible for the characteristic and also phenotypic characteristic. Already I told you phenotype means what? Visible, something which is visible. For example, a hair color or height or eye color. So the resemblance of the parent. So the what's visible to our eye, whether the child is tall or having blue eyes or curly hair. So these are all visible. So what is externally appeared as what we say is phenotype, phenotypic character. So the character from the parent is taken to the child through the help of DNA. DNA is a carrier. So the particular character from parent to a child is carried, that is from one generation to other generation. The informations or the character are carried with the help of the DNA only. So this Mendel experiment we discussed, isn't it? He used the pea plant and he denoted the genes with the letter capital T, small t. That is T, capital always denotes the dominant characteristic, small t, recessive characteristic. All this we discussed. Okay, just recall that. And very simple matter here. The genes are responsible for carrying the information from one generation to the other generation. And then one more term you should know, locus. Locus is nothing but specific position at which each gene is present on the chromosome. Yes, I told you this is a homologous chromosome. So one is paternal, other one is maternal. Okay, so a set of homologous chromosome is given for you, and here you can notice the black dot. So this is gene. Okay, so the chromosomes are made up of genes, and particular character is denoted by a certain gene. See here in the top, if the gene is present in the top for one pair, in other pair also, that is maternal also has the gene in the same position. Okay, for example, if this gene denotes the eye color, here also it denotes the eye color. The position is same, right? Here if you see that this denotes the appearance of the hair texture. Okay, just for example, I am telling you, if here a gene is present representing the hair texture, means same way the other homologous pair also contains the gene in the same position. So the position never changes for the homologous chromosome. So here locus is what? The position where the gene is present is the locus. Just the example I am showing you here. So this is a 2 mark or 1 mark it will be asked for. So locus is what? Specific position at which each gene is present on the chromosome. And also tell you that during the cell division only these chromosomes are visible, and this chromosome contains the DNA, that is genetic material, that is gene, which is carried from one generation to the other generation. Not only for 2 generations, it will be carried for many generations. Waldeyer, he only first coined the term chromosome in the year of 1888. T.H. Morgan, in the year of 1993, he got the Nobel Prize for determining the role of chromosomes in the heredity. These are all one words. Next, let's see the structure of a chromosome. This is chromosome, okay? So let's see the detailed structure of chromosome. The chromosomes are thin, long and thread-like structure. You can see the chromosome here. The chromosomes are thin, long, thread-like structure. Consist of 2 identical strands. You can notice this is one strand, this is one other strand. So it is made up of 2 identical strands called sister chromatids. So this is one sister chromatid and this is one sister chromatid. Okay, so sister chromatids are joining and forming the chromosome. And the sister chromatids are held together by centromere. You can notice in the middle, centromere. So sister chromatids are joined with the help of centromere. Okay, so what is present in the middle of the chromosome? Centromere. And next, chromatid. Chromatid, it's made up of spirally coiled thin structure. You can notice the chromatid here, okay? This is a chromatid. Now, one chromatid is shown for you here. It is made up of spirally coiled thin structure. You can see the spiral coiling inside, okay? This is called chromonema. What is this? Chromonema. Black thread-like structure is chromonema. And this chromonema consists of number of bead-like structures. You can notice the red dot. So these are bead-like structure along its length, which are called as chromomere. So bead-like structure is what? Chromomere. Thread-like structure is what? Chromonema. Where it is present? In the chromatid. And these chromosomes are made up of DNA and RNA. Okay, deoxyribonucleic acid and ribonucleic acid. These chromosomal proteins are also present, that is histones and non-histones are there, which are responsible for providing the structural support for the chromosome. And also some metallic ions are also present. Metallic ions are also present in the chromosome. And then this chromosome consists of the following regions. This is very important, children. Please listen carefully. Primary constriction or centromere. You can notice the primary constriction here. So the chromosomes are meeting at a point. The two arms of the chromosome are meeting at a point called as primary constriction. This is a region where spindle fibers attach to the chromosome. So this is the chromosome. The arms of the chromosome are getting attached to the primary constriction. The two arms are getting attached to the primary constriction. So during the cell division, spindle fibers attach to the chromosome. For this you must recall the cell division. See, this is during the cell division, various phases are undergoing. Here I am showing you two phases, metaphase and anaphase. Okay, so during the cell division, chromosomes are visible, and these chromosomes are coming closer to the middle, that is equatorial region. That otherwise you can say it as metaphase plate. They are getting arranged in a metaphase plate. When it is getting attached, you can notice the spindle fibers are getting attached to where you can notice this line. These lines are spindle fibers. Okay. So this spindle fibers are getting attached to the middle part. What is that I told you? Chromosomes. Spindle fibers are getting attached to the chromosomes where in the primary constriction or centromere. So this is the centromere. Previous slide also I can show you. Centromere. This is the centromere. See, this is a centromere. So in this area, centromere only, the spindle fibers get attached. You can see the spindle fibers are getting attached to the centromere. This region is very important. And next, what is happening? In the next phase, what is happening? The chromosomes are getting separated. Only then two daughter cells can be formed, right? So this chromosome is getting separated. How? The spindle fibers will pull the chromosome towards itself. So chromatids get separated. Okay, so what's the point to be noted here? The spindle fibers get attached to the centromere. See, the region where spindle fibers get attached to the chromosomes. Hope you understood, children. Next, secondary constriction. First, primary constriction I explained to you. Now, secondary constriction, you can notice it here. Otherwise, you can say it as nucleolar organizer. It is present at any point of the chromosome. Secondary constriction is present in any point of the chromosome, and it is present in some of the chromosomes. It is not must to present in all the chromosomes. It is present in some of the chromosomes. It is responsible for formation of the nucleolus in the nucleus. Inside the nucleus there is nucleolus, isn't it? So for the formation of nucleolus, what is helpful? Secondary constriction, or that's why it is named as nucleolar organizer. And next part, telomere. Telomere, you can see this area. This is telomere. This is at the end of the chromosome. Okay, this is in the end of the chromosome. Each extremity of the chromosome has got a polarity. So each chromosome in the extreme end it has got a telomere and it has got a polarity. Polarity means this word is not new for you. Polarity. Magnet has got the polarity, isn't it? So around the magnet there is a magnetic power, which we say it is polarity, isn't it? So that what's the use of this polarity means this prevents it from joining the adjacent chromosome. So one chromosome will not be attached or touching the other chromosome. What is preventing that? This polarity. So this polarity, which is responsible for that telomere. Okay, and this maintains and provides stability to the chromosome. This telomere gives stability and maintains the stability to the chromosome without touching the other chromosomes. The polarity will be maintained here. Okay, this is a part telomere over. So three parts they have explained you. One is primary constriction, secondary constriction, and now telomere is over. Next, satellite. Satellite, you can notice this area, satellite. This is elongated knob-like appendage. Can you notice? It is knob-like structure. Okay, so this knob-like structure you can view in the fan regulator or door handles, etc. Okay, so this such a structure is the satellite. It is present at one end of the chromosome, and only in some of the chromosomes it is present. Such chromosomes are called sat chromosomes. Where the chromosome where the satellite is present is called sat chromosome. So remember this clue word, S, triple S. So satellite as well as secondary constriction are present in some of the chromosome. So this is secondary constriction, satellite are present in some of the chromosome. It's not compulsory to be present in all the chromosomes. With that, the structure of chromosome is over. So these are the important parts, children. Remember, primary constriction, secondary constriction, telomere, then satellite. Next term what you should know is karyotype. Karyotype, very simple term only, ma. So in any living organism, either it is an animal or plant, the number of chromosome is constant. So it is constant. The number never changes. For example, if you take a rice plant or Rhiza sativa, the number of chromosome is 24. If you take a human being, Homo sapiens, the number is 46. Okay, and chimpanzee 48, dog 78, likewise. So the number of chromosome changes and it is fixed for each organism. For example, for us only 46, 23 from paternal, 23 from maternal. So totally 46 chromosomes are present. So, in human, each cell normally contains 23 pair of chromosomes, out of which 22 pairs, 22 pairs are autosomes, and the last pair, 23rd pair, is allosome. So, allosome is nothing but sex chromosome. You can notice in the structure, 1, 2, 3, likewise, the structure is going on. You see, these are all chromosomes. Okay, chromosomes are arranged in this way from bigger to smaller. Okay, so the structure will be like this. 22, totally 23 pairs are there, isn't it? 22 pairs are autosomes, which are defining the body characteristics. 23rd pair alone, allosome. Why? Because that is a sex chromosome, which is helpful for the reproduction. So important one word, 23rd pair alone, allosome. In sexually reproducing organism, whether it is a male or female parent, that is a sexually reproducing organism, that is sex chromosome. In body cell, the chromosomes are generally occurring in pair only. All are diploid in nature. In body cell, all the chromosomes are diploid in nature. 2n number of cells will be present. See, except the reproductive cells. Okay, so this is the reproductive cell. 2n number of cells is present in all the cells, body cells, isn't it? And only this reproductive cell alone undergoes meiosis cell division. Body cells undergo mitosis cell division. Reproductive cells undergo meiosis cell division. And if you take for male, sperm is a gamete. Sperm is a gamete for male, and it is haploid in nature. Single N is present. It is not diploid, haploid in nature. Why? Single set of chromosome only will be present. In gametes, single set of chromosomes is present. That's why this for male sperm as well as for female egg. Okay, for female egg as well as for male sperm, these are reproductive cells and it is having only n number of cells, so it is haploid. It undergoes what cell division? Meiosis cell division. And here, if you take karyotype, the number, size and shape of the chromosome is the karyotype. In this, so previously I taught you this only to know the haploid cell is present in the reproductive cell. So in other body cells are diploid in nature. You can notice it here, ma. The last pair of cell alone is sex chromosome, which is haploid in nature. All the other are diploid in nature. So this arrangement is shown in the karyotype structure. Okay, so this is a karyotype, ma. The number, size and shape of the chromosomes are denoted in the structure. Okay, and that structure, diagrammatic representation of an organism or species is what we say, it is ideogram. The diagrammatic representation of karyotype of a species is ideogram. Important one word. What is ideogram? Diagrammatic representation of a karyotype of a species. You can see for this insect, this is a karyotype structure which is shown for you here. That is ideogram. Okay, so this consists of all the metaphasic chromosomes arranged in a homologous pairs. So this chromosomes are arranged with the homologous tracks. Diagrammatically it is represented. And how it is arranged? According to the decreasing length, thickness, position of centromere, shape, etc. So decreasing length, thickness, position and all. So I showed this is bigger in size. As it goes on, the size is getting reduced. Based on the size only it is arranged. And finally, sex chromosome alone placed at the end, at the last. You can notice it here. So totally 23 pairs are there. 22 are left in the top. Last 23rd pair alone sex chromosome. That's why it is left in the last. Other than that, remaining chromosomes are all arranged based on the size only. Decreasing size, decreasing length. Okay. So all the 22 pair of chromosomes are autosomes. The last pair alone we say it as allosome. So with that, the structure of chromosome karyotype, the karyotype explanation is over. So I have explained you the structure of chromosome and karyotype in this video. Thank you, children.